Hello friends, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning platform. Today we learn about physiologic barriers. These physiologic barriers are also called as biochemical barriers. This include general conditions and some specific molecules. The physiologic barriers that contribute to innate immunity include temperature, pH, and various soluble and cell-associated molecules. Now, we'll see how the condition temperature prevents infection or how it plays an important role in innate immune response. Many species are not susceptible to certain diseases simply because their normal body temperature inhibits the growth of the pathogens. Chickens, for example, have innate immunity to anthrax because their high body temperature inhibits the growth of the bacteria. So higher temperature of the chickens make them resistant against anthrax. Next, we'll see how the pH will bring about the resistance against the infection. Gastric acidity is an innate physiologic barrier to infection because very few ingested microorganisms, consumed microorganisms can survive. Infected microorganism can survive the low pH of the stomach contents. One reason newborns are susceptible to some diseases that do not afflict or do not infect adults is that their stomach contents are less acid than those of adults. So newborn babies or newborns are very susceptible to infection because their stomach contents are less acidic than those of the adults. When the same microorganism infects or ingested in the stomach of the adults will not cause any infection. Coming to various soluble and cell associated molecules, here we'll be learning about lysozymes. Interferons and complement proteins. First we'll see the lysozyme. The lysozyme, a variety of soluble factors actually contribute to innate immunity. Among them, lysozyme, which is a hydrolytic enzyme found in the mucous secretions and in tears. Lysozyme is present in mucous secretions and in tears, is able to cleave the peptidoglycan layer of bacterial cell wall. Whenever there is bacterial infection, the bacterial cell wall can be digested or the bacterial cell can be damaged by this lysozyme. So this lysozyme is a hydrolytic enzyme. It is produced in the mucous secretions and it is rich in the tears. So this will prevent the bacterial infection. Next is interferon. Interferons comprises a group of proteins. These are group of proteins produced by virus infected cells. Specifically, these interferons will be produced by virus infected cells. Among the many functions of the interferons is the ability to bind to the nearby cells and induce a generalized antiviral state. So these interferons are also called as antiviral proteins. So these are produced by virus infected cells, and they bind to the nearby cells and they induce antiviral state in the virus infected individuals. The next is the complement. These are otherwise called as complement proteins. These complement proteins are group of serum proteins. These are group of serum proteins that circulate in an inactive state. These complement proteins are inactive proteins. They circulate in inactive state. A variety of specific and non-specific immunological mechanism can convert these inactive forms of complement proteins into an active state. So in the body, these complement proteins are inactive serum proteins, and in order to get converted into active state, they require a specific or non-specific immunological mechanisms. So these will convert these inactive complement proteins into active complement proteins. So these active complement protein, they can damage the membranes of the pathogenic microorganisms either by destroying the pathogens or facilitate their complete clearance inside the body. So these complement proteins are serum proteins. They are circulating in the body, gets activated by specific or non-specific immunologic reactions, and when they are activated, they can damage the membranes of the pathogen. Either they kill the pathogen or they completely remove the infection or the pathogens from the body. The complement may function as an effector system that is triggered by binding of antibodies to certain cell surfaces, or it may be activated by reactions between complement molecules and certain components of microbial cell walls. So activation of the complement protein may take place either by the interaction with the antibody on this specific cell, or they may be activated by either another complement molecule, or they may be activated by the components of microbial cell walls. So these three reactions, either by antibody or by other complement protein or by the microbial molecules, will activate the innate or adaptive system to bring about the resistance. And these complement proteins are very degradative in their function. Many of the molecules involved in innate immunity have the property of pattern recognition, the ability to recognize a given class of molecules which are present on the specific microorganism, and they are absent and never found in multicellular organism. So these unique molecules will be recognized by the innate immunity, and that molecule we call it as the pattern recognition receptor, or this process is called as pattern recognition. The molecules with the pattern recognition ability may be soluble like lysozyme and the complement components, as I told earlier, or they may be cell associated receptors. So these can recognize the specific molecules which are present on the surface of the microorganism. organisms. So this class of receptors include toll-like receptors, will be usually referred as TLRs, toll-like receptors. So these toll-like receptors usually they recognize the lipopolysaccharide layer, lipopolysaccharide, or it is in short form it will be called as LPS, found on the gram-negative bacteria. So biochemical barrier or physiologic barriers include certain conditions and the biochemical mediators or the small molecules. So the conditions like temperature and pH will bring about the resistance against the infection, or cell associated or the molecules, simple molecules can also bring about the resistance. That includes lysozyme, an hydrolytic enzyme, complement proteins, or your interferons. So these are the molecules may be soluble, important role in avoiding the infection. Apart from that, on the surface of the cell we'll find certain pattern recognition. This specifically recognizes the specific microorganism and they inhibit the infection. So such receptors, example is the TLRs, that is toll-like receptors. They recognize the lipopolysaccharide molecule present on the gram-negative bacteria. I hope you understood biochemical barriers or physiological barriers involved in the bringing about the resistance in innate immunity. Thank you.